Hello, my name is Emily Rainge, and I am a high school student in the SIMI program. This summer, I worked in Dr. Khanda's pharmacology lab, studying the building blocks of amyotrophic lateral sclerosis, also known as ALS. ALS is a 100% fatal progressive neurodegenerative disease that affects nerve cells in the brain and the spinal cord. In the US, there are more than 20,000 people who have the disease at a given time. My lab's question is, how can we lower the fatality rate of ALS? Well, we think certain target proteins within muscles could play a part in answering this. This is why my lab decided to study the protein TDP43. TDP43 is an RNA-binding protein that translocates to the cytoplasm during stress, where it forms cytoplasmic RNA protein complexes, known as aggregation. Disease-linked mutations in TDP43 enhance the ability of TDP43 to form protein aggregates in vitro and in animal models. It binds to and regulates the splicing and stability of numerous RNAs encoding proteins involved in neuronal development, synaptic function, and neurodegeneration. So, a loss of these essential functions is an attractive hypothesis regarding the role of TDP43 in neurodegeneration. While I was in the lab this summer, I did cell cultures with E. coli, did protein purifications through centrifuging, ran gel tests on proteins to check their purities, and ran saturation transfer difference spectroscopy experiments on the compounds to check binding points of compound to different strands of protein and drug-target interactions. We have found that if we can identify where compounds are binding to TDP43 strands, we will be able to identify the compounds that inhibit TDP43 aggregation. The questions that I am now left with are, is there a genetic hereditary component to ALS? How could a clinical test for mutant TDP43 proteins be invented? What other neurodegenerative diseases is TDP43 linked to?